everyone, my name is Veronica Bubay, and today I will be sharing with you my 2022 summer research project with the Phelps Lab, where my project focused on designing and confirming a model for predicting diffusion behavior through granular hydrogels. Before we dive into things, I wanted to tell you all a little bit more about myself. I'm originally from northern New Jersey, and I'm currently a rising senior at the University of South Carolina, where I'm studying biomedical engineering and art history. I'm on track to graduate next spring in May of 2023. Polyethylene glycol, or PEG hydrogels, have emerged as a highly influential platform in tissue engineering for both their heightened biocompatibility and easily tunable mechanical properties. However, the viability of encapsulated cells is threatened by the limited diffusion capabilities of these bulk gels, particularly their nanoscale porosity, which impedes sufficient nutrient diffusion. Granular hydrogels, on the other hand, formed from closely packed hydrogel microspheres, offer a really promising advantage as their packing creates cellular scale void spaces that both increase diffusion and cell migration while still maintaining the favorable biophysical characteristics of bulk PEG hydrogels. With that in mind, this work aims to create and confirm an efficient computational model to compile granular hydrogel variables to determine rates of nutrient diffusion. For our lab's purposes, we chose to use COMSOL Multiphysics 5.6 to accomplish these goals. To do so, I created a block geometry with side lengths of 150 microns that were formulated to contain perfectly packed microspheres, with a diameter of 31.2 microns. This number was used as it's proven as ideal by my mentor Adrian's previous research. The main goal when modeling this geometry was to maintain spacing equal to 0 microns. This means that our microspheres are touching throughout the model. This allows for the pore size, also known as the void space, to be directly related to the diameter of the microspheres. I then used the transport of diluted species physics module to perform diffusion as it allows for user control of gel properties, including the microsphere diameter and thus pore size, block length, initial solute molecular weight and concentration, as well as hydrogel and void space diffusion coefficients. Then, to create a more realistic model than embodied randomized microsphere packing, open source Blender was used to simulate randomly initialized spherical rigid body particles falling through a funnel and into the same block geometry. This was inspired by work done by Riley et al. The final random pack geometry was then exported into the same COMSOL model built to predict diffusion. Studies were then run using a protein with a molecular weight of 155 kilodaltons and a concentration of 10 micromolar for an interval of 1 hour with 2-minute time steps. These models shown on this page are taken at the t=4 minutes time point. We can see here in the obtained concentration gradient for both the random and perfect models that diffusion within the pore space occurred faster than within the microspheres themselves. This is in line with what we predicted based on our granular hydrogel properties. We also compared these models to nutrient diffusion profiles through both bulk gels and solution. And, as shown in the figure, the model is consistent with an improved rate of diffusion in granular hydrogels compared to bulk gels. To confirm our model, we used a tritzy-dextrin invasion assay, where we placed 155 kilodalton molecular weight tritzy into one well on an Abiti chip, and our gels in the other well, and tracked the diffusion profile using a Leica confocal microscope. The image on the slide was taken at the t=4 minutes time point. Moving forward, we plan to extend our experimental validation of the model by running experiments at various other solute weights and concentrations, as well as at other measured diffusion coefficients. This model can even be applied to islet encapsulation models to predict in vitro behaviors before approaching the experimental setting. Ultimately, this model can be used to indicate optimal pore sizes in granular hydrogels to facilitate the most efficient nutrient diffusion profiles. I'd now like to take a moment to acknowledge all those who have shown me their support this summer, including Dr. Phelps, my mentor Adrian, and the whole of the Phelps Lab for creating such a welcoming environment to grow and learn as a researcher. I'd also like to thank the SURF program here at UF, especially Mike Nazareth, for curating such an exciting summer research experience. Interacting with students from so many different fields of engineering introduced me to new fields of research that I otherwise would not have learned about. I'm now looking forward to furthering my research career by pursuing a PhD or MD-PhD within the field of biomedical engineering. I'd like to say thank you again to my mentors, the SURF program, and fellow SURF students, particularly my roommates, for making this such a memorable summer for me. Please don't hesitate to reach out, and thank you for your time listening today. Bye!